Myself, Dr. Arundathi HA, here with you all to help you navigate through the subject of microbiology in various PG entrance exams. So today, let us discuss a few PYQs in today's PYQ series, where we will try and understand the concept pertaining to each and every PYQ in details. So let us move on to our first question. So here we have a child presenting with a history of high fever, recurrent seizures, headache and neck stiffness. So all these are the features of meningeal irritation. So there is meningeal irritation which is resulting in all these particular symptoms. And the CSF analysis reveals low glucose, elevated protein and also increased PMNs. And the CSF culture shows pleomorphic gram-negative bacilli. Question is, which of the following is true? So, which is true regarding the causative pathogen, whether it exhibits alpha hemolysis, on-sheet blood agar, differential motility, NLF colonies, or satellitism. So the first thing is, as they have given a clear history of meningitis, we will need the analysis of biochemical markers if the options had a mixture of bacteria, tuberculosis or viral etiology. But this is not the case. So here the case is they themselves have given that they saw a gram-negative pleomorphic bacilli in a culture after growing and after say doing a gram stain. So now we should concentrate only on the bacterial causes of meningitis and understand meningitis with respect to the same. So now let us look into the various causes of bacterial meningitis. We can understand the causative agents under the following headings in terms of morphology: gram-positive cocci, gram-negative cocci, gram-positive bacilli, and lastly gram-negative bacilli. So under gram-positive cocci, we have two gram-positive cocci, that is Streptococcus pneumoniae and Streptococcus agalactiae, which are the gram-positive cocci causing meningitis. Gram-negative cocci only one, that is meningococci, that is Neisseria meningitis. Is a gram-negative cocci causing meningitis. GPB again only one gram-positive bacilli, that is Listeria monocytogenes. Listeria monocytogenes is the causative agent. Among gram-negative bacilli, we have pleomorphic bacilli. Pleomorphic means they appear as gram-negative coccobacilli as well as gram-negative bacilli. And a pleomorphic bacilli is Haemophilus influenzae. And other gram-negative bacilli, others are Escherichia coli or Klebsiella. So these are the causative agents according to the morphology. As in the question, we have the morphology of the bacteria which is given. What will be the additional hints in the question pertaining to each of these organisms? In case of Streptococcus pneumoniae, so this is the commonest one causing meningitis, and usually in the history, the gram stain picture will be given, or a gram stain picture explanation will be given, saying they are gram-positive cocci which are in pairs and which are capsulated. So gram-positive cocci in pairs and capsulated is more towards Streptococcus pneumoniae. Streptococcus agalactiae, they are gram-positive cocci which are present in chains, and what is most important is they are the one who causes neonatal meningitis. So neonatal meningitis is caused by Streptococcus agalactiae. Then gram-negative meningitis, along with all other symptoms of meningeal irritation, a classical case of meningitis. Here the patient will have a history of skin rashes, where the skin rashes either in the form of petechial or purpural lesion will be seen. So this will be the important feature in case of Neisseria meningitis. And what are they? They are gram-negative cocci again which are in pairs and they are capsulated. So these are capsulated gram-negative cocci in pairs. Then Listeria monocytogenes. So here usually there will be history of elderly with patient with a history of meningitis, or it can also cause meningitis in neonates like that of Streptococcus agalactiae, or in case of immunocompromised patients. So Listeria is a commonest agent in all these, and it mainly presents what is called as differential motility. So Listeria, what does it produces? It produces what is known as differential motility. And what does this differential motility mean? So differential motility means they are motile at a lower temperature, that is 25 degrees Celsius, and at a higher temperature they are non-motile. So as the temperature increases, what we feel? We do not feel like moving around, right? So the same way, even Listeria does not move. They are non-motile at a higher temperature. They do not feel like moving around, and they are motile at 25 degrees Celsius. And the other one is pleomorphic gram-negative bacilli, which is H. influenzae and others. So this is in toto about the hints pertaining to the important causative agents of bacterial meningitis. So moving on to the question again. So here we have pleomorphic gram-negative bacilli. So when this pleomorphic is given, what is the causative agent? We all know this causative agent is H. influenzae. So this is a case of H. influenzae meningitis. As these options are pertaining toward mainly the cultural characteristics of this organism, we will study about H. influenzae in detail. So H. influenzae is a fastidious organism, and this organism requires two growth factors. One growth factor is factor V, and the other one is factor S. This factor V is not present on black agar, whereas factor X is present. On chocolate agar, however, both the factors are present. Therefore, where do they grow well? So they grow well on chocolate agar, but not on blood agar. So to make them grow on this blood agar, what do? So we take a blood agar plate. Then we streak an organism in the center which releases factor 5. Factor 5 is not there in blood agar. Which is this organism? We use Staph aureus. So we streak Staph aureus in the center on blood agar, and then our H. influenzae like this. And the growth of H. influenzae next to the streak line of S. aureus will be larger as compared to when they move towards the periphery. So the larger colonies nearer to S. aureus streak line, and they becomes smaller as they move to the periphery, and this is called as satellitism. So satellitism is this particular phenomena seen in H. influenzae. Okay. So now let us look into the options. It exhibits alpha hemolysis on sheep blood agar. What did I tell? They will not grow on sheep blood agar well because there is no factor V. Shows differential motility? No, it is a non-motile bacilli. So which shows differential motility? What is the one which shows differential motility? We have Listeria monocytogenes, which shows differential motility. They form NLF colonies on McConkey agar. No, they will not grow on McConkey agar. And lastly, yes, they exhibit satellitism around S. aureus colonies. This is the correct option. Fine. Let us move on to the next question. So here, an elderly female presented with meningitis. Again, it is a case of meningitis, and her CSF sample shows gram-positive bacilli. So we directly have the diagnosis here, gram-positive bacilli. And what is true? So what is true about the organism? So let us go back to this one. And what is the organism which is gram-positive bacilli? We have Listeria monocytogenes. So our option here is Listeria monocytogenes. And what is true about its motility? It is motile at 25 years. At lower temperature, it is motile. Non-motile at 37 years. Because as temperature increases, it does not feel like moving. Yes. So the correct option is Listeria monocytogenes, motile at 25 degrees but non-motile at 37 degrees Celsius. As we have got the right answer, we need not waste time in checking the other answers. So moving on to the third question, a 23-year-old male presented with a history of fever with 4 days history of myalgia, headache, retroorbital pain. There is no signs of bleeding, and on examination there are rashes on the body. What is the next appropriate step in management of the patient? What do you want to do? What is the next test you want to do? Vidal, NS1 antigen, IgM ELISA, or a rapid influenza antigen test? So firstly, we will have to diagnose this case. What is the diagnosis of the case? By this particular history of myalgia, headache, retroorbital pain with a comment on bleeding and maculopapular rashes. It is a case of viral hemorrhagic fever caused by dengue. So dengue is classified as what? Dengue fever with or without warning signs, and severe dengue. Okay, so therefore there need not be bleeding always. So need not be bleeding always. There may be no bleeding, or there may be rashes on the body. So what is the diagnostic test of dengue? So this choosing a diagnostic test depends on what is the history of the patient, from how many days the patient has fever. So here the patient is having fever for 4 days. What happens is, in the initial days, the patient body, the virus will be actively replicating. Say up to 5 days, you can identify what? So only antigen. So you can identify only antigen up to 5 days, because immune cells requires time to get activated and produce antibodies. So for more than 5 days, if there is trace of more than 5 days, then you can go and detect antibodies. So which is the antigen which is detected in dengue virus? The antigen is non-structural antigen 1, that is NS1. And the antibody after 5 days history, the first antibody which appears is IgM. This will be followed by appearance of IgG. IgG will not appear first. First one is always IgM. Mother is the first guru. Like that, mother IgM is the first fighter in our body. Okay. So the options here, let us look for NS1 because the history is 4 days. So yes, B is the option. So moving on to other options also, where do we do Vidal test? Vidal test is done in enteric fever. Both the typhoid as well as paratyphoid is under enteric fever. In IgM ELISA, we would have chosen this option if the history was of more than 5 days. For first 5 days, you cannot detect IgM antibody. And yes, you can detect it either by ELISA or by a rapid CAR test, which is called as immunochromatographic test. Rapid influenza antigen, the case scenario itself is not matching with influenza. So moving on to the next question, a young male presented with a history of dysuria and urethral discharge. Dysuria plus urethral discharge, the diagnosis, you see urethritis. The patient is presenting with urethritis, and a gram stain of the purulent discharge is shown below. What is the treatment choice for this infection? So my suggestion is, when an image is given, it is an image based question, and you are very much clear about the image, you are 100% sure what this image is, then don't waste your time reading the entire question, but go and read the last sentence of the question. If you're 100% sure, you do not have even 1% doubt about the image. Okay, this is fine. This is having only 3 lines, but sometimes the question itself runs for 6 to 10 lines. And then how to save time? Then you can save time if you are thorough with the image. So this is an image where we are seeing the gram-negative cocci which are within the cell. So intracellular gram-negative cocci which causes urethritis. It is nothing but your Neisseria gonorrhoeae. So Neisseria gonorrhoeae is the one which causes urethritis, which is called as gonococcal urethritis. And what is the treatment of choice? As the question is about treatment, we have list of antibiotics here. The treatment for Neisseria gonorrhoeae is mainly your 3rd generation cephalosporin, either ceftriaxone, which is the first drug of choice, or it is cefixime. So cefixime or ceftriaxone is the drug of choice. And if at all you also think about a concomitant infection, along with gonococcal infection, you have non-gonococcal urethritis like a chlamydia, so then you add azithromycin in treatment. Okay, so you have gonococcal as well as chlamydial infection, azithro is added along with ceftriaxone. But as it is a confirmed case of gonococcal urethritis, we are going with only ceftriaxone. So the option here is ceftriaxone. If in this place cefixime was there, we would have chosen even that. Okay. So the next question, which of the following toxins acts by a mechanism depicted below? So here we have a mechanism, and the options are diphtheria toxin, botulinum toxin, Clostridium difficile, and step pyrogenic exotoxin. So one thing, if an image like this is given and you have no clue about what the image is, how can we solve this question? You go and analyze each option. So you know diphtheria toxin, how it acts. It inhibits elongation factor 2, then it inhibits protein synthesis. So this is how it acts. Do we have EF2 here? Do we have protein synthesis? No. So this is not the answer. Okay. So this is how we can analyze and answer the question if we are not perfect or if we don't have any idea about the image that is given. Fine. So now, but here let us solve by analyzing the image here. Here in image we have antigen presenting cells. So how does this antigen presenting cells act? So this AP cells, the antigen will be taken up by this AP cells and they will be presented to T cells. So how is it presented? So APCs bears what? It bears MHC class 2 molecule, and this MHC class 2 molecule bearing APCs will present to T helper cells. So class 2, just draw a line here. This is how you remember class 2 is T helper cells. Class 1 is for T, the other one, that is CD8 T cells. Fine. So T helper cells, that is MHC class 2. So then this T cells will activate the B cells and the B cells produces antibodies. This is the normal mechanism that we see. But what is happening here? So this is the antigen which has not gone to APCs. No, it has not gone to APCs. Instead of going to the APCs, it is directly going and activating the T cells. So this antigen which is acting like a superman is called as superantigen. So it is acting like a superman where it is directly activating T cells, where it goes and binds to what is called as variable beta region of the T cells. So can you see here V beta, variable beta region of T cells, which activates the T cells directly and resulting in polyclonal activation of B cells. Okay, this is how superantigen works. But however, to select the options, we should also know the examples of the superantigens, right? How do we remember superantigens? A very easy mnemonic is we will be like supermans in chemistry lab. Okay, so that's how we remember. So chemistry is the mnemonic that we have for superantigens. So we will be so excited in chemistry labs. We were during PU and we used to act like superman and superwoman. So similarly superantigen. What is C? C is CMV, H is HIV, E is EBV. So these are our viral superantigens. Viral followed by, let us take up a fungal superantigen, that is Malassezia. So Malassezia furfur is our fungal superantigen. I is for me. Okay. Then ST is for both staphylococcal and streptococcal superantigen. Staphylococcal, what all we have? We have three. That is toxic shock syndrome toxin, enterotoxin, and exfoliative toxin. There are three toxins which are superantigen. Streptococcal pyrogenic exotoxin. So pyrogenic exotoxin is a superantigen. Whereas R is for rabies, where the rabies nucleocapsid is a superantigen, and Y is for Yersinia. So Yersinia, both pseudotuberculosis and your Yersinia enterocolitica. So is it clear? So how do we remember superantigen? We used to act like superman and woman during chemistry lab, right? So chemistry is the mnemonic. C for CMV, HIV, EBV, Malassezia for a fungal agent. Then ST for staphylococcal and streptococcal superantigen. R for rabies and Y for Yersinia, that is pseudotuberculosis and Yersinia enterocolitica. Shall we search if any of these are here? Diphtheria, no. Botulinum, no. Clostridium, no. Streptococcal pyrogenic exotoxin, do we have it here? Yes, so streptococcal pyrogenic exotoxin is there, and this is our answer. So this is about the superantigens. Let's move on to the next question. In which of the following organisms genetic material is adenine not equal to thymine? Yes, an interesting question. So as soon as we see this adenine and thymine, what should we know is A is equal to T and G is equal to C where? In DNA. Whereas in RNA, what happens? In RNA, A is equal to U, that is uracil, right? So what do this simply mean is, in which of these organisms the genetic material is not DNA, that means it is RNA. This is the simple meaning of the question. So here let us see. So E. coli, Escherichia coli, what is it? It is a bacteria. And what do we know about bacteria? All bacteria have DNA as genetic material. So HIV is virus, we don't know because virus may have DNA or they may have RNA as genetic material. Herpes, the same thing holds good for herpes because it is virus. Human cell, human mitochondrial cell, all human cells, we have DNA as genetic material. So to solve this question, we should know what are the DNA viruses and which are the RNA viruses. So how do we remember DNA and RNA virus? I'll just touch upon DNA virus because all others will be RNA, right? Because DNA are less number of viruses, easy to remember. So DNA viruses are basically happy virus. So they are happy virus where H is hepadna. Hepadna comes your hepatitis B virus, then your herpes viridae. Herpes many comes under this herpes viridae, that is your herpes simplex virus, varicella zoster virus, EBV. Then you have CMV among the most important one. Then you have adenovirus. Adenovirus is adeno itself. Then you have papilloma virus. Papilloma itself and a parvo. Then we have parvovirus. Then you have pox viridae, where under pox, chicken pox is not pox. What comes under pox? Your smallpox is pox, and along with that, molluscum contagiosum virus is smallpox. Why is for us? We are happy because we know all the DNA virus. So hepadna is hepatitis B, herpes many and herpes virus, adenovirus, papilloma virus, that is human papilloma virus HPV, parvovirus, that is your parvovirus B19, pox virus, smallpox, and molluscum contagiosum. All these are DNA virus. The rest of all are RNA virus. So these were the two which we were doubtful about. So herpes virus is a DNA virus. So HIV, did we see HIV here? No. So HIV is the one which is a RNA virus, and yes, the genetic material is adenine not equal to thymine. Okay. Let us look into the next question. A patient with PO is suspected to be suffering from Brucellosis. His serum samples undergoing standard agglutination test shows negative agglutination at 1:20 and 40 dilution, but positive at a higher dilution of 80 and 160. What causes the negative result in the first two dilutions? The first thing is, don't break anything your head anything about the first two lines, because the question is not related to that. The question is related only to the dilutions given here. So what is present here and what are the options? Okay, we will look at the options: incorrect antigen, antibody, excess of antigen, excess of antibody. So let us solve this question. The first thing that we should know is what has happened here is at a lower dilution. So at a lower dilution, the test is negative, whereas as the dilution was increased, that is at a higher dilution, the test becomes positive. Okay, and this standard agglutination test, we are testing to see for antibodies in the patient's blood. Okay, so what happens in a lower dilution? Antibody will be more. So this is something like, take a glass and add sugar granules to prepare a lemon juice. So what you will do, you will put sugar and then you will put water. So initially, when you put only little water, you will be able to still see the sugar granules, or the sugar will be more. So this sugar is equal to antibody. Okay. Then what you will do is you will add more water, and now all the sugar granules have dissolved nicely, and this tastes the best. Okay, the taste is the best here. Whereas what you will also feel, let me add some more water to it, and you added more water, so the sugar content has decreased so much that it is not at all tasty. Water is more. So let us compare this to the antigen and antibody quantity. So here where sugar was more, so here where the sugar was more is equal to antibody, this is called as a zone which is called as Prozone phenomena, where the antibody is excess and the result will be negative. Okay, this here where water is more, so here what will be excess? Antigen will be excess. Water is equal to antigen. Antigen will be excess. Again, there may be chances of negative result. Okay, sometimes. Whereas this that is where there is equal quantity of antigen and antibody. So antigen is equal to antibody. It is the best, right? So this is called as zone of equivalence, and here there is formation of lattice. Lattice formation will be there, that is perfect agglutination reaction, perfect antigen antibody reaction will be there. Okay, so here where the dilution is less, so what is excess in the dilution? So 20 and 40, but in dilution increased, it became positive means. So when dilution was less, what is this? What is more? Antibody is more. Let us search for the same, and this is the answer. Okay. But let us also analyze other answers. Incorrect antigen and antibody. If at all the antigen or antibody was incorrect, never ever it would have become positive. Okay, fine. So this next option is antigen excess. We know that as dilution increases, when we are looking for the antibody in serum, as we are adding more and more saline, no, antibody, antigen excess will not cause positive result. It would have caused the negative result. So the answer is the antibody excess. Okay. Fine. So the next one, a female presents to the OPD with complaints of thick, curdy white vaginal discharge for 2 days. Which of the following can be used to identify the likely causative agent? So this is the one which is repeated many times, where the classical history is that of curdy white vaginal discharge. Always remember this curdy white is equal to another C, that is Candida. So Candida albicans is the agent which causes vaginal candidiasis, where there will be curdy white vaginal discharge. So what we should know about this Candida is other C's also. That is, so very first C of Candida is you can speciate it on what is known as CHROM agar. So CHROM agar is an agar which produces different colored colonies, say light green, dark green. So different species of Candida produces different colors on this CHROM agar. So that is mainly for speciation. Next is cornmeal agar. So cornmeal agar is the next C, where cornmeal agar is used to elicit one more C of Candida, that is chlamydospore. So chlamydospore is a round thick-walled spore which is seen in Candida, and you can pronounce the production of chlamydospore by growing on cornmeal agar. So speciation is mainly done by growing on what? Cornmeal agar, one. And the other way to confirm Candida albicans is what is known as germ tube test. So germ tube test is a test where there will be production of germ tubes arising from the yeast cell. That is germ tube test positive in Candida albicans. Let us see a saprolus dextrose agar. We just used to grow it. Okay. CHROM agar is, this is the answer we want. Bird seed agar, no. Did we come about a bee anywhere here? No. So brain heart infusion agar, no. This does not help in differentiating the species of Candida. So next one, a sewage worker is, this is also important for us, presents with jaundice, abdominal pain, headache, retroorbital pain, conjunctival suffusion, and blood in urine for the past 5 days. What is the investigation of choice? Wheel Felix, Paul Bunnell, Vidal, and microscopic agglutination test. Okay, so first we should diagnose this. So we should know what is this condition, and then we can choose the test of choice. Okay, so a classical history of icterus, that is jaundice, along with all other, with this particular conjunctival suffusion is drawing us towards the diagnosis of leptospirosis. So leptospirosis usually it presents in two manifestations, that is two stages. One is an icteric phase and the other one is icteric phase, or the severe disease, okay, which is also called as Weil's disease. Fine. So it is this particular case is that of the icteric phase, where the patient is having jaundice, abdominal pain, and all these aches and pains, and conjunctival suffusion because of conjunctival hemorrhage. So conjunctival hemorrhage and also blood in urine, which is the characteristic feature in case of Weil's disease. And again, another hint is sewage worker, because this is a zoonotic infection which you get from contaminated water, where the water is contaminated with the animal's urine. So usually it is the rat's urine contaminated water which results in leptospirosis. So we have done the diagnosis. So what is the test of choice for leptospirosis? We tell it as MAT. What is MAT? Microscopic agglutination test, and agglutination test which is done in a microtiter plate. So shall we search for the same? Yes, so we have it here. This is microscopic agglutination test done for leptospirosis. So let us try and understand other options also. Where do we do Vidal test? Vidal test, I already told you, it is done in enteric fever. So we do Vidal test in enteric fever. Where do we do this Paul Bunnell test? So we do this Paul Bunnell test in EBV infection. And what type of test is it? It is a heterophile agglutination test. So it is a heterophile agglutination test. Another heterophile agglutination is the Weil-Felix test. Where do we use it? We do it in rickettsial infection. So in rickettsial infection, we do Weil-Felix, Paul Bunnell in EBV infection, enteric fever, Vidal. And what is our answer? So this is the answer which is correct, that is microscopic agglutination test. So the next question, a 30-year-old male presents with complaints of persistent cough and weight loss. So he was HIV positive, there is persistent cough and weight loss, and the fungal culture of the sputum was negative. Okay, so the suspected fungal etiology because he was HIV positive, and GMS, that is Gonorrhea methanomen silver stained picture of the sample is shown below. What is the most likely causative organism? So again, if you are very much 100% sure of the image, you can skip the whole history. But if you are doubtful even for 0.5%, please look at the history. So what is the most likely causative agent? So this is a case of HIV patient and immunocompromised patient. So the common fungal infection in this immunocompromised patient, that is HIV patients, is pneumocystis. So pneumocystis jirovecii. So pneumocystis jirovecii infection is the commonest infection. Okay, so we have spelling here only if we have doubt in the spelling. So pneumocystis jirovecii infection is the commonest infection which happens in this immunocompromised state, and most so it is given the hint, a clear hint is in the form of a picture. So Gonorrhea methanomen silver stain, where this pneumocystis jirovecii shows what is called as crushed ping pong ball appearance. Crushed ping pong ball appearance, also referred to as crescentric cysts. So we have crescentric cysts which are present. So these are the two morphologically explained forms of this particular pneumocystis jirovecii. So all these are crushed ping pong ball appearance. Here we have crescentric forms which is seen. And yes, can we search the same? Yes. So pneumocystis jirovecii, which produces crushed ping pong ball, and this is one of the fungus which does not grow on the culture media. It does not grow on the culture media. So if we have to try and understand all of this, because all of this are again what? So these are dimorphic fungi. So even if we know the morphology of all of this, we can come to the conclusion. Histoplasma. So histoplasma, what do we have? We have tuberculate macroconidia. Tuberculate macroconidia is present in histoplasma. How this is macroconidia? This is how it looks. So these are tuberculate macroconidia. Coccidioides immitis, cocci. So what you have? You have this pose, and within that you have endossus. So these are looking like cocci, right? Coccidioides, you have cocci-like structure. Pneumocystis jirovecii we studied here. Then paracoccidioides, P is equal to another P, that is called as pilot wheel appearance. So pilot wheel appearance. So this is how a pilot wheel appears. So these are the morphological features of all other options. Moving on to the next question. A group of friends ate pastries from a local shop late at night. Okay, so we have two hints here. One is pastries and late at night. When did they develop any symptom? early next morning, so late at night and early next morning, so it should be less than say about 6 to 8 hours, less than 6 to 8 hours. All of them develop vomiting. So this is most important. What can be the probable causative organism? We have list of organism. So this is a case of food poisoning because after eating something they have got the manifestation, and food poisoning which happens less than 6 hours, it is classically because of two bacterial causes. One is Staphylococcus aureus as the most common cause. The other one is Bacillus cereus. So these are the two causes. If we have a history saying that the time between eating or consuming the food and the symptoms is less than say 6 to 8 hours. Okay, but what is more towards how do we come to a conclusion here? Cirrus is C is equal to carbohydrate rich food. So C is equal to carbohydrate rich food. That means you have fried rice, which is carbohydrate rich, or you have noodles, or you have pasta, or any starch rich food. So these are the ones which gives rise to Bacillus cereus, where you can either have vomiting or diarrhea. Whereas Staph aureus is mainly with the dairy products. So dairy products, that is milk or milk products, are the one which gives rise to Staph aureus related food poisoning, again manifesting as vomiting because they stimulate the vagus nerve resulting in vomiting. Okay, fine. So here the pastry is will have cream and all, which is made up of milk. So you mainly this is a dairy product, and therefore it should be Staph aureus. Let us search. Yes, so the answer is Staphylococcus aureus directly. So this is a easy kind of question. So next, an army man presents with itchy rash all over the body. On microscopic examination, a red pigment, very nice, a red pigment producing fungus with pencil-shaped macroconelia is produced. Which of the following is most likely organism? We have list of organisms here. So in this particular case, itchy rashes all over the body. It is nothing but Tinea corporis case, a fungal infection, dermatophytic infection. And what is more important in the history is, one is this image. Okay, we have an image. If we are thorough with the image, we can dive into the answer. The second one is they have also commented on its cultural characteristic, which produces a red pigment. So for us, what is important is this red pigment is produced by two fungi. One is Trichophyton rubrum. Rubrum is red pigment producing trichophyton. The other one is Telleromyces marnafi. So Telleromyces marnafi is the one, and the other one is Trichophyton rubrum, which produces red color pigment. So we can directly dive into the answer here, where we have the option of Trichophyton rubrum. Okay, because Telleromyces does not produce the characteristic history like this, and we do not have it in option also. Okay, but let us say that this particular thing was not given in the question. Then how do we come to the diagnosis? Then our diagnosis depends on our mastery on the various species of Trichophyton. So they have given pencil shaped macroconelia. What are the different shapes of macroconelia of different Trichophyton species? So very first one, let us take this Trichophyton rubrum. So rubrum, as I told, this is the answer. So it should be pencil shaped, right? So pencil shaped in case of rubrum. So violation means there is a lot of violence. So violence, what it produces? It produces distorted hyphae. Distorted hyphae is produced in case of violation. Nothing characteristic feature in macroconelia, but because of violence, there will be distorted hyphae. Then Sean-Laney will produce what is called as favic chandelier. So favic chandelier shaped macroconelia. How do we remember? Let chandelier. Why do we use chandelier at home? For show purpose. In a local language, for show purpose, we use chandelier, and show is Sean-Laney. Okay. Then we have tonsurans, T for thickened macroconelia. So the tonsurans produces thickened macroconelia. Okay, we have one more which is most important, that is Trichophyton mentagrophytes. So this dicophyton mentagrophyte produces the spiral hyphae, which is important one, and also it produces cigar-shaped macroconadia. Mentagro R is the most important for us here. R for spiral and R for cigar-shaped. Okay, so mentagrophyte, spiral hyphae with cigar-shaped macroconadia, tonsurans T for T, that is thickened macroconadia. Sean is show. For show, what do we use at home? We use chandelier. So fabric chandelier type microconadia. Violaceum. So due to violence, the hyphae has got distorted. So distorted by a hyphae. And rubrum, we should remember. So rubrum B, you can just remember like this. B, make it P, is your pencil-shaped macroconadia. So moving on to the next question, a newly joined band member developed lysotomy stool. This is again an easy question. The causative agent act on which of the following receptor? First, we should know the causative agent, right? So as soon as we utter the word rice watery stool, as soon as we utter it, we should notice Vibrio cholerae. So Vibrio cholerae is a causative agent of cholera which causes rice watery stools. And how does this Vibrio cholera act? Because it is the question is all about how does it act. Okay, various receptors are given. So it produces cholera toxin which has A and B subunit, and the B subunit binds toward GM1 ganglioside receptor. Please remember, it is number one causing rice watery stool. So it acts on GM1 ganglioside receptor, which results in increase in CAMP. And what does the CAMP do? It causes secretion of both NaCl and water into the intestinal lumen, resulting in rice watery stool. Fine. So where did it act? It is number one agent causing rice watery stool. So it should act on what? GM1 ganglioside receptor. So the next question, a 40-year-old male presents with cough and breathlessness. On taking his history, he gives a history being profession of a driver and diabetic. A diagnosis of pneumonia was confirmed and histopathological investigation revealed a right angle branching, like how you see here. So right angle branching, what is the likely infection? Okay, a case of pneumonia, and here the risk factor is diabetes, and looking at this image, so this is going in favor of what? This is going in favor of mucormycosis. So in mucormycosis, the hints in the question are usually the risk factor for mucormycosis is increase in iron concentration, which mainly happens in diabetic ketoacidosis. So we will have a history of diabetes or a patient on iron therapy, but more commonly diabetic ketoacidosis is the risk factor. The next hint in this question is right angle branching, where mucormycosis, the microscopic feature of mucormycosis is, the very first thing is they will have the broad hyphae like this. Do you see any septate here? No, there are no septate, therefore a septate hyphae. And the branching will be a broad angled or wide angled or right angled branching. Okay, so microscopic feature of mucormycosis is what? You have broad hyphae. What about the septate? It is aseptate. There is no septate. And what is the other thing? The branching thing. The branching is either wide angle, it is obtuse angle, or it is the right angle. Okay. With this particular mucormycosis, what else should be differentiated is, mainly we should differentiate it from aspergillosis, which is almost the same, but the risk factor can be anything else, neutropenia, steroid therapy, also not necessarily diabetes. Okay. And microscopic feature, if they had given the microscopic feature, what you will see is a narrow hyphae is the characteristic feature of aspergillus. And they are septate. You will find septate in the hyphae, and they show acute angle branching. So there will be acute angle branching, something like this. Okay, how is it looking? It is giving a finger-like appearance. So it gives finger-like appearance because of acute angle branching. So here it is a clear case of mucormycosis where there is broad hyphae, no septate, and right angle branching. Aspergillus we have finished. finished it. Histoplasma, in the previous question I said, please remember T like this. T is the main thing, tuberculate microconidia. And Candida, we know they are gram-positive, or let me say, they are budding yeast cells with pseudohyphae. Okay, so they do not show this true branching and all. They are not the molds, they are yeast-like fungi. So, moving on to the last one, the 15th question. Which of the following disease is caused by Coxsackie A16? We have list of diseases here. Fine. Coxsackie virus is one of the virus which is most commonly touched upon concept. So, let us understand in detail about Coxsackie virus. So, Coxsackie virus is basically divided into two groups, that is group A and B. A, we have serotypes from 1 to 24. A is always the first and it will have more, and B is 1 to 6. Okay. What are the common infections in A? Let us begin with A itself. One is aseptic meningitis. So we have aseptic meningitis caused by Coxsackie virus. What are the serotypes causing it? It is mainly caused by 7 and 9. Okay, I'll tell you how to remember later. The second one, it causes what is known as herpangina. So herpangina is also called as febrile vesicular pharyngitis. So here the patient will have history of fever and there will be vesicular lesions. The ulcerative lesions will be there on the pharynx. Okay, what are the serotypes causing it? All even numbers till 10, that is 2 to 6. Please remember, it is not 2, 6, it is 2 to 6. For convenience sake, let us remember as 2, 6, 8 and 10. Okay, so these are herpangina causing serotypes. The third one is, it can also cause nodular pharyngitis. There will be almost history of pharyngitis, but there will be no ulceration in the pharynx. That is nodular pharyngitis caused only by 10. So nodular only by serotype 10. And fourth one, which is one of the commonest one, it is hand, foot, mouth disease. So hand, foot, mouth disease, where there will be vesicular lesions in the oral cavity, also in the soles and palms. So that is hand, foot and mouth disease, which is mainly caused by Coxsackie A16, and along with that it is also caused in combination with enterovirus 71. Okay. The last most important one is your hemorrhagic conjunctivitis. Hemorrhagic conjunctivitis, which is caused by A24 along with enterovirus 70. Okay, so how do we remember it? Aseptic meningitis, A is first, so first letter. So it is the odd numbers. Okay, so odd numbers are 7 and 9 among the list given. So at least we should by heart this list. If we get confused, we can apply this theory. I did not get any other easier way to remember. So herpangina, all positives, that is 2 to 6, 8 and 10. Nodular pharyngitis, O, so O is the important thing here, and 10. HFMD, so how many letters are there? 4. Okay, so how many causative agent? 2. So 4 is written twice. So what is it? It is 16. Okay, so it is A16. After 6, what is the number after 6? 7. And 1 again, 71, enterovirus 71. Usually we get confused with HFMD and hemorrhagic conjunctivitis. So this is how we remember HFMD, 1, 2, 3, 4 letters. How many causative agent? 2. So therefore write 4 twice. 4 into 4 is 16. So A16. After 6 you have 7, enterovirus 7. Again 1 transfer here, that is 71. Then we have hemorrhagic conjunctivitis. Hemorrhagic conjunctivitis is A24. The 16, the most important is 16 and 24. If we know about 16, the left is 24 and enterovirus 70. So this is about Coxsackie A. Coxsackie B, what are the important ones it causes? It can also cause aseptic meningitis. All the serotypes can cause. More common are 1 to 5. And what B is most common causative agent of is called as Bornholm disease. So Bornholm disease, also called as pleurodynia, also called as devil's grip. So devil's grip is there will be stabbing like chest pain, and that is called as devil's grip or Bornholm disease, again caused by all the 6 serotypes of B. It can cause myocarditis, it can cause pneumonia, but what is one more important thing is pancreatitis, which may result in diabetes mellitus, and the serotype is B4. Okay, so pancreatitis is B4, Bornholm disease almost everything, and it is also called as devil's grip because of stabbing chest pain. So let us look for the answer for this. Coxsackie is 16. Okay, so it is hand, foot and mouth disease. Fine. So yes, we studied about it. And yes, we will stop at this particular stage where I hope you all thoroughly enjoyed this particular session. Looking forward to meet you again and again. Thank you very much.